So I will talk today about biological processes in the atmosphere, which is probably not really a topic that is that popular in the atmospheric sciences, but I will show you what effects these aerosol particles can have on the composition of the atmosphere, but also on clouds. So in my outline, I will show you, for those who are not that familiar with it, what is the role of aerosol particles, any particles in the atmosphere, and what is so particular about biological particles. So I'll give also a brief outline of our MOBGA project, which is modeling biologically driven processes in clouds, or called MoBiDiC. And then I make it clear, so why do we care about these biological particles and processes? And I will focus on some of the processes like cell generation and growth, biodegradation, and also ice nucleation on biological particles. And throughout my talk, I will actually focus on bacteria as microorganisms, because we just have most data and we know most about them. And so in my summary on conclusions, I will summarize what do we know so far, and probably more importantly, what do we don't know, and where do we need more data and more constraints to include it in models. So this is just a schematic about the role of aerosol particles in the atmosphere. So in general, there are two effects. One's the aerosol direct effect. So aerosol particles that are emitted from all kinds of different sources can interact with light, so with sunlight, and depending on their chemical composition, they can either absorb light, so warm our atmosphere, or they can scatter light, so they cool it. The aerosol particles can be either emitted directly in form of particles, or the majority of the particles is actually formed in the atmosphere from condensable gases that either make existing particles grow or nucleate new particles. So more complicated than this aerosol direct effect is the aerosol indirect effect. So particles can take up water vapor, and if the conditions are right, then they can form cloud droplets. And some of these particles can even initiate freezing. So last week we heard in the seminar here from Anna a lot about mixed phase clouds, so clouds that contain liquid water and ice. And so of course, it depends on the meteorology and on the thermodynamic conditions, but it also depends what kind of particles have below the clouds and what kind of particles form ice and which don't. And because these interactions are so complex of particles and with clouds and the different cloud phases, so liquid or ice, aerosol-cloud interactions still count as one of the largest uncertainties in our current estimates of global warming. So now, what does biology have to do with it? So first here, a short summary about the size range. So if I talk about aerosol particles, what is the size range I'm referring to? So here, the most abundant particles are likely sulfates, soot, and organics. So they are somewhere in the nanometer scale, or maybe up to a few hundred nanometers. Then we have mineral dust and sea spray, which are somewhat larger. And then on any of the particles, if they are above a certain threshold of a few hundred nanometers, for example, they can act as these cloud condensation nuclei on which cloud droplets form. When cloud droplets grow, they become rain droplets and fall down, or they form ice. So we spend several orders of magnitude here. And down here, I roughly indicated the scale for biological particles, which are a subsection of all the atmospheric particles. So what are biological particles? Well, I mean, when we wrote the project proposal, we had no idea how popular suddenly the viruses become. But of course, we all heard about a lot about viruses last year and still going on. But also there are a lot of bacteria, fungal spores, and pollen in the atmosphere. And they have different size ranges depending on the different subtypes of these particles. And also they have different concentrations depending on emission sources. So these are rather average concentrations. And overall, these biological particles make only a very, very small percentage of the total particle loading in the atmosphere. So it might be somewhat more than a few percent locally, but globally, it's certainly a very rather small fraction. But why do we care about them at all? So they have very unique biophysical chemical properties interacting with light and water in ways that other particles don't. They're dispersed and transported in the atmosphere, so in particular in clouds. So that means if they are transported, they also come down eventually from the atmosphere and affect biodiversity on a different location as where they were emitted. And unlike other particles, they can be also modified by biological processes. So we know a lot about chemical and physical processes, but these biological processes that are obviously unique to biological particles are much more poorly constrained. And they can initiate very particular transformation processes like enzyme reactions, biotransformation, and biodegradation. And I will touch on some of these processes a little bit in my talk. So for me, the atmosphere is really an interdisciplinary lab. So I've been working on chemical processes in the atmosphere for the past 20 years now, and I've focused a lot on cloud chemistry. So soluble gases that are emitted from many different sources can dissolve in cloud droplets, undergo chemical reactions there in the water, as opposed to in the gas phase. And then they can modify the initial cloud condensation nuclei and form larger particles with a different composition. Of course, also outside clouds, particles play a role as condensation nuclei, or they can be modified by gas phase reactions. So far, atmospheric models contain atmospheric chemistry and atmospheric physics. And for sure, we don't understand all these processes yet completely. However, as compared to atmospheric biology, these models are fairly well constrained. Because atmospheric biology, any of these processes are, if at all, very, very poorly included in models. And that was really the idea for our MOPCA project for Moby Dick, that we say, well, we take the data that is available and put it into models and see how important individual processes are. So this is basically the overview of Moby Dick, that we want to include lab studies, which have been done in Clermont-Ferrand in our group for 10, 20 years. Then there's a strong effort on field measurements. They have been also been done for several decades. I will talk about them very briefly, but we want to connect the pieces by modeling and understand the processes, test these models, and then finally, we want to put them on larger scale models, regional or global scale, to really understand, to make conclusions about their global role. So I can tell you right now, we are not quite there yet, but I will show you the different pieces we have addressed so far in our project. So what can happen to a bacteria cell that gets emitted from different ecosystems? So obviously different ecosystems, whether I have a forest or whether I have the ocean or other ecosystems, they emit different bacteria and different at different concentrations. But eventually they get into the atmosphere and might get involved into a cloud droplet. And actually bacteria cells like water. So for them, they are the least stressful conditions in the atmosphere, because in an aerosol particle, the water is very limited. So it's a very harsh condition for them, highly concentrated. So they try to attract water. And so cloud droplets are probably the best conditions they can encounter in the atmosphere. So while they are in a cloud droplet, several things can happen to them. So they can either grow or multiply, or they can be dormant, so nothing happens to them, but they're still alive, or they can die. So no matter what happens to them, so cloud droplets will evaporate, and eventually these particles will be deposited to the surface. So, but for us was a question, so how much can they actually grow? To what extent can actually the bacteria mass increase while a bacteria cell is in the atmosphere? And so we termed this increase in mass secondary biological aerosol mass. So, and we performed a very rough estimate how much that might be, just to get an idea whether it's worthwhile to put more effort into that to constrain parameters. So basically, we broke down the process into several steps. So if we start with a bacteria cell, we know the cell concentration. So how many cells are there in clouds on average? So these are measurements that are more or less routinely performed here, for example, here in France. Then we know from lab studies, so from the literature, what are the growth rates of typical atmospheric bacteria. And then, of course, we have to constrain the time where this can happen in the atmosphere. So how long does bacteria cell probably spend in clouds while it is in the atmosphere? So basically, that is our first approach, how to estimate this increase in bacteria mass. So basically, we did estimates for all the different parameters. So we know how the mass of a cell, we know the cell concentration in cloud water. Then we know the cell growth rates. Of course, it's a large range because every cell, every bacteria strain or bacteria type has a different growth rate. And finally, we can estimate also the time a particle spends in the atmosphere. We have models to estimate that mostly depends on the size of the particle. But also we can estimate how much time spent of that time is actually in cloud. So it's only a fraction of clouds. So on average, it's about 15% of the time a particle spends in clouds, depending on the region. But it's certainly not 100%, but it's not negligible. So basically, we based our approximation or our estimate on a previous study, on a global model study by Burrows et al., where the authors actually looked at bacteria concentrations above different ecosystems. So I tried to color code this table by the same colors here, for example, desert in yellow and so on. And obviously, there are different concentrations because different ecosystems emit different amounts of bacteria and different types of bacteria. So that was the basis for our estimate, but we wanted to extend it and said, so for the predominant bacteria from each ecosystem, so based on literature data on these predominant bacteria, we could estimate this growth rate. And also we could estimate based on satellite measurements, for example, for cloud cover, what is really the area that is covered by clouds above the different ecosystems, above the seas, above the deserts, and so on. And so having this data, we could just put it together by the same equation I showed you before. And then we scale it by the area each ecosystem covers on Earth. And we could come up with production rates as a function of the different ecosystems. So here are the production rates in the plot. On the y-axis, we have the production rates in teragrams per year. And on the y-axis, we have the different ecosystems. And so we did two different estimates. The one was really the ecosystem dependent parameters. And then another estimate was just an average value for global parameters. And we get two different values, which are 3.7 or 0.7 teragrams per year. I mean, of course they differ, but they're roughly on the same order of magnitude. And what we really wanted to see is whether it compares somehow to the direct emissions of primary bacteria before they start growing, before they are emitted to the atmosphere. And so it's roughly on the same order of magnitude. So our conclusion was that actually this secondary biological aerosol mass production might be an important source for biological mass or bacteria mass. So that was the first process I wanted to discuss. The second one is biodegradation by organics by bacteria. So we all know that there are a lot of organic compounds in the atmosphere, both in the gas phase or also in particles or in cloud water. And they are very detailed chemistry models that describe their loss processes or oxidation processes in the air and in water, so in the cloud phase. But the group here in Clermont-Ferrand, for the last 15 years or so, they have not only measured bacteria cells in cloud water, but they also took them to the lab and measured the biodegradation rates for different bacteria strains that they found in cloud water, but also how quickly they consume actually the organic compounds in water. And so that is one of the major objectives also of our MOBGA project, that we say, so how important is biodegradation as compared to chemical losses? Because I have these very detailed models for cloud chemistry, and now we wanted to add the bacteria, the biodegradation. However, it's different. It's not a simple addition, because we know that in cloud droplets, we have basically all chemical compounds because they're exposed to the gas phase. So basically chemistry happens in all cloud droplets. However, only 1 out of 1,000 or 10,000, depending on the air mass, droplets contains a bacteria cell. So how does that play out overall as towards the overall loss rate? So in order to address that, we use the box model, because of course, the importance of this biodegradation for a single organic compounds really depends on many different factors, which is how reactive is it chemically in the gas and aqueous phase? How soluble is it? I mean, if it doesn't partition to the aqueous phase, probably the aqueous phase process is not that important. And then, of course, how quickly does it get consumed by different bacteria? And so these are the new data we have from our lab. And these are data we have in cloud chemistry models or multi-phase models, or gas and aqueous phase models available. So we use the box model, just like shown here. We have many droplets, we have a gas phase, organic compounds and oxidants are in equilibrium or are exchanged between the two phases, and 1 out of many droplets has a bacteria cell. So we only compare the chemical and biological processes. So we do not consider wet deposition, because that would be a different process we cannot address really with a model like this. So these are the results. So at least some of them. So we looked at the total loss of this organic compound by chemical plus biological process, so biodegradation. And we only did a simulation time of about 15 minutes. It sounds very short, but we could easily scale it up. But all what we are interested in is just the ratio between chemical processes and biodegradation processes. And so the total consumption of the organic compound is very small. So the total consumption after 15 minutes is only 1.6%. So it's very small. But what you can see here is on the one axis, it's the chemical reactivity in the aqueous phase. On the other axis, so pH is the Henry's law constant, which is a measure of the solubility. And it's exactly what I would expect, that if the reactivity is very high, if the solubility is very high, a compound is very efficiently consumed. So now this is a total loss. So this chemical plus biological. So if we now look at the fraction of biological processes, we get actually this figure, the bottom figure. So the scales are different. And the pattern is different. You see immediately that we have a maximum at a solubility of roughly 10 to the 5 Henry's law constant, so intermediate solubility. But if you compare the numbers, it's roughly where the total loss is about 0.2%. We have 0.06% by biodegradation, so it's roughly 30%. So our conclusion was that for these compounds that have an intermediate solubility, the biodegradation might be most important, might be add up to something like 30% of the total loss. So models that do not include that underestimate the degradation of these compounds. And these are compounds like formic acid or acetic acid, which are really major cloud water constituents. So I mean, this was an interesting result just in terms of numbers, but we wanted to understand why do we have this weird pattern, which is not that intuitive as compared to chemistry. Well, here we understood it, so highly soluble, highly reactive. Of course, aqueous processes are important. Here we have this different pattern, but that has something to do with this multi-phase system, that we only have the bacteria in 1 out of thousands of droplets. And what happens there is that the loss in this one special droplet with the bacteria is so efficient that a lot of the organics are taken up from the gas phase, and then the gas phase gets depleted, so the concentration of the organics becomes very small. And then from the other droplets, we start evaporating these organic compounds. So we have a redistribution because we have this very, very efficient sink in this one special droplet with the bacteria. And the very, very soluble compounds, they don't want to evaporate. So they stay in so it's really hard for them to evaporate. So it's more the intermediate soluble compounds that will evaporate. And therefore we see this different pattern. I mean, it's very interesting in terms of the numerical consideration, but it's really bad news for modelers because in many cloud chemistry models, it is assumed that all droplets are the same. And if you assume that all droplets are the same, you never get a pattern like this. You don't see that really the intermediate soluble compounds are the more important ones as compared to the very soluble ones. So that definitely requires some parameterization or new description, how to describe it also in larger scale models where we do not have all these individual droplets. So so far, I talked about microorganisms and how they change aerosol properties. And let me just quickly summarize what I said so far. So there are two major processes, so cell growth and generation, forming secondary biological aerosol mass. We think it might be important, it might be an efficient source of biological or bacteria mass in the atmosphere under the good conditions, ideal conditions. And also bacteria can degrade organic compounds in cloud droplets. And we think that for compounds like formic or acetic acid or some compounds with similar properties, it might be an efficient sink in addition to the chemical processes we have included in models. Of course, our assumptions so far are very, very simple, and we have very simple models, highly idealized models. And also the assumptions, especially for the cell generations, are very rough, but at least they tell us it might be worthwhile to dig into these processes a little bit deeper and to get more data to constrain these conditions. We also made the large assumption that all these processes are restricted to clouds. There is some evidence, I think, that processing can also occur in wet aerosols. So because aerosol particles outside of clouds also have some water, they're never dry. But so far, we don't really have any data we could really put into a model. So the second part will address microorganisms and cloud microphysics. So this is a schematic now on the left side here. I showed you now in different versions. So aerosol particles, any aerosol particle, but also bacteria, for example, can make cloud droplets. And some of them will freeze. And ice particles will precipitate very readily because they are usually larger and heavier than cloud droplets. But a lot of bacteria will not freeze, so they will be transported over long distances. And eventually they might be removed by wet deposition, or the cloud evaporates and then we have dry deposition. But that means this traveling of the bacteria in the atmosphere means that also the bacteria are transported and will get deposited somewhere else far away from the emission sources. This freezing of bacteria has been known for many decades. And so bacteria are really special because they freeze at fairly high temperatures, so above -10 degrees. Other particles freeze at much lower temperatures, at -30 or -20, depending on their composition. So in order to understand this, we have to understand, or we want to understand, why bacteria are so good ice nucleators. So very briefly, for those of you who work on aerosol-cloud interactions, it's not news, but for the others here, just a very quick rundown. So what makes a particle a good cloud condensation nuclei? So it's particle size. They have to be able to take up water, and to some extent also the curvature of the particle, which is related to the surface tension. So we have a fairly good handle on that. So if we know a composition of a particle, we can basically say whether they are a good CCN or not. What makes a particle a good ice nucleus? Well, it has something to do with the particle surface, to a smaller extent, the particle size. And so now the question, why do bacteria form ice? Well, there is a theory out there that they want to precipitate. So the atmosphere is a very harsh environment, so limited water, they're exposed to all kinds of oxidants and to UV light. The particles shrink and grow, and so there are very rapid changes in pH and dilution. So it's not really ideal conditions. So once they're in the atmosphere, they try to attract water, and then they freeze to get precipitated and to get removed again. So that is one of the idea why bacteria, so just from the biological point of view, freeze. But how do we describe it in models? Well, this is an overview from a German group where they put together data from lab studies. And on the y-axis, it's the parameter NS. So I don't want to explain in detail what it is, but it is related to the ice nucleation ability. A high value means it's a good ice nucleus. Low value means it's not freezing that readily. And of course, freezing is a function of temperature. And so in this green shaded area, you see the bacteria, and these are really the only ones that freeze at above -10 degrees. So what we put in current cloud models is really an equation like this. So the number of frozen particles, if you know how many particles we have in total, the number of frozen particles is a function of this ice nucleation ability and temperature. But this is very empirical. So there is no way for us, if we have an unknown bacteria, to say, well, where would it fall into this NS range? So we try to do another approach and think about why are bacteria so good ice nucleators? Because we have this idea, if you understand the why, if you identify the property, then we can also extrapolate. So it's fairly well known that it's not the full bacteria cell that freezes, but it's really protein molecules that initiate the freezing. And only a small number of all bacteria cells initiate ice nucleation. And because they might have these special proteins that freeze. So from the microbiology community, it's fairly well known that proteins form aggregates. Unlike other ice nuclei, for example, a dust particle, they don't do that. There's a compound there. So aggregate means we have monomers, we have small molecules. If the concentration gets higher, they make larger molecules. So something like oligomers, or then if it gets larger, maybe polymers. And these large molecules have a really high order and are very well structured. And so that is very good for ice to form, because also ice crystals are obviously very well structured. And so our idea was, if we can parameterize this aggregation, maybe we get a step further to understand or to parameterize ice nucleation on proteins, and with that on bacterial cells somewhat better. Because, I mean, if we go back to this figure, it is not something we only came up with because we think it should be like that. We also discussed this idea with several groups who do lab experiments in physical chemistry labs, and they see indeed this concentration effect. The higher the concentration, the more likely is this aggregation, the more likely is the ice nucleation. So this has consequences for cloud chemists or for cloud models, because if you think about a particle that has a protein, becomes a cloud droplet. Of course, a cloud droplet in the beginning is very small. That means the concentration is high. Cloud droplets start growing higher in the cloud, but also the temperature drops higher in the cloud and eventually initiates freezing. So it might be that the freezing patterns that are described by current cloud or by ice nucleation models might change if we have this concentration dependence. So this is work in progress. We are not quite there yet, but we think we can improve current ice nucleation parameterizations on proteins. So this is an overview from a large sensitivity study my postdoc Minghui Zhang did, which also summarizes the interactions of biological particles with clouds. So there are two effects. There's a warm clouds, so forming cloud droplets, and there are the cold clouds or mixed phase clouds that also include ice. And there are different parameters, so diameter, hygroscopicity, and surface tension, for example, for CCN. This ranking by no means is new. However, so it applies to any particle, but we also thought about, okay, what happens if I have a biological particle like a bacteria cell? What processes can change these parameters to an extent that might be important and should be therefore included in models, this aging process or this modification process. I talked about the cell generation. It might be enough to make a bad cloud condensation nuclei to a good one. There are a lot of chemical processes and physical processes which are described to some extent in models, but also there are processes like biosurfactants. So bacteria produce something to attract water. So we haven't really explored that, but that might be possibilities. We also want to explore to see how biological particles interact with water. For ice, the ranking is different again. So these sensitivity studies have been done before, but they have never been done really with the aspect of how can biological processes really modify biological particles. So again, so that is not only affecting clouds, but it's also affecting the residence time of these biological particles in the atmosphere. So now I showed you this schematic before, but I come back to it because now I showed you some examples what we have done in our project on lab studies, data compilation, and process models. Now we'll talk briefly about ambient measurements and field experiments, because we are in the very lucky position that so just 15 kilometers west of Clermont-Ferrand, it's a Puy de Dôme, and there's the observatory, which is a global atmospheric watch site. That is an observatory that is managed by the physics department or meteorology department of our university. And for several decades, they have routine measurements of aerosol clouds and precipitation, and of course, meteorological parameters and so on. So we have a wealth of data from this station and also some other stations nearby, where we could just test our models. And on top of these chemical and physical parameters, there are also a lot of biological parameters which were measured by our group. And so one of the setup could look like this, or did look like this in the past. So we know that air masses at the Puy de Dôme arrive from many different directions. So this is from a study a few years ago, where the clouds or the air masses were classified from their direction, and whether something comes from the ocean or comes from the north, from Paris or other directions, of course, the composition, the chemical composition is different. So they all arrive here, and we have a good idea where they come from when they arrive. So they can get collected here, then they travel further, and we did an experiments at Ida Lake, so which is about 10 kilometers or 15 kilometers away, and collected rain samples. And then, of course, the cloud might exist further and travel further, or the air mass gets further transported. But at least we get this traveling time of the air mass and of the particles. And so here are some first results. And just an example, what can be measured? So it's a total bacteria concentrations, specific strains, so different types of bacteria, and also in rain versus cloud samples. And also we can measure how much is really precipitated, how much is removed from the atmosphere. And questions we can address with measurements like this is, for example, is the microbial population in cloud and rainwater the same? Well, it's not. And then the next question, if it's not, then why do certain bacteria really partition to the one or to the other? And here is an overview from our PhD student, where she looked at the partitioning of certain bacteria genus or types, and in cloud and in rain. And you can see there is a difference. And so the differences can be quite substantial. So clearly, either some bacteria like to partition to the droplets that participate, or some other mechanism is going on that changes these the partitioning or the concentrations in cloud water and rainwater. And even more so, is the bacterial activity in these two different water samples the same? Well, it's not. So you can see it here. Again, we have cloud water and rainwater and the ratio between the two. It might differ quite a bit. So these are the biological variables in the right part of the plot. And so, but we should also keep in mind, rainwater is not only cloud water. It might also wash out the air below the cloud, or it might have something to do with the bacteria that they partition to rainwater versus cloud water. So we are not sure yet, but it's something very interesting. We could also explore with models to see, can we really find the process that can explain this different partitioning? So with this brief overlook of our field measurements, I want to summarize my talk. So Moby-Dick is really putting pieces together, putting pieces of evidence together of the role of biological aerosol particles. I mostly talked about, only talked about bacteria, but the same I said might also apply to other biological particles, but we have even fewer data for those than for bacteria. So so for none of the processes, we really have the full picture yet, but I mean, we did this very rough estimate for cell generation on a global scale, and we think it might be important, but of course we need to constrain it with more data or more specific data. Biodegradation, we haven't really put on a global scale yet, or because we cannot parameterize it yet, this one special droplet in a cloud. But we also think that it might contribute to the loss of several organic compounds. We are trying to improve currently the ice nucleation parameterization for the proteins, bacteria proteins. We have this very interesting data on the different partitioning between cloud and rainwater samples. So we could probably explain it with the models, but we haven't really put the pieces together yet. And so we have done very extensive sensitivity studies to identify processes and parameters that might affect biological particles enough to get included in clouds or not. So that will give us guidance what other data and what other processes we want to look at to finally get the full pictures for this whole suite of processes. So now I want to conclude, and also so in the spirit of the Make Our Planet Great Again program. So what is the importance of biological particles for global change? Well, I cannot really put a number on it yet, but we think they can modify aerosols by biodegradation and can also grow sufficiently to increase biological mass. Of course, there's a cloud formation and the ice formation on bacteria, biological particles, but we need really have a deeper understanding of the bacteria-dependent properties that causes ice nucleation. But I also would like to turn it around and say, what is the importance of global change for biodiversity? So if we look in the future, I mean, emissions of microorganisms will change with changing temperature and land use. And I'm sure we will have several talks in the seminar series about it. And also changing cloudiness will affect the residence times and the transport pathways of the microorganisms. And so, yeah, I want to conclude with this call. So if you know someone who might want to work on these topics, or if you are someone, just let me know, because we are still looking for 2 postdocs who could continue this work. And with that, I finish and take any questions.
Okay, thank you very much for this excellent talk. Yeah, I'm sure there are many questions. You can use the chat, type questions in the Q&A, or if you are one of the panelists, you can also just raise your hand. I have several questions, and I will therefore start. So the first question I have, can you also calculate the CO2 release that the bacteria produce? And so how much is it in relation to the global carbon cycle? And yeah, okay, and then I'll ask another one.
We did calculate it. It's actually very, very little. So I forgot the exact percentage, but it's actually not that much. I mean, of course, biodegradation eventually forms CO2, but it's actually very, very small, just simply because the total bacteria mass or the microorganism mass is not that high in the atmosphere.
Mm-hmm. Okay, and the second question I have is, you said, okay, you think bacteria might want to cause ice formation to get out of the clouds, right? And
that's
that would
one of the theories. I mean, yeah. We always think that any biological process has a reason. You know, they try to optimize their living conditions.
But yeah, but I mean that assumes that that the cloud phase, so to say, is part of their normal life cycle if they would adapt to it. So do you think that's the case, that there are really bacteria who are a part of whose part of the life cycle is to be in the clouds? Then that would also lead to the question: okay, do they adapt and create ice nucleation devices? And for me, the question is also, I've worked on sea ice biology before, and in that milieu, the organisms want to avoid ice formation, because if ice forms in the cell, it actually deletes the cell unless it's the ice is in a glass state. So they would also need to do something to have the cell protected from the ice formation to survive the ice formation itself they would induce. So it's quite complicated. So how
I'm not saying that this is the only reason why they make ice. I mean, there are also lots of antifreeze proteins that try to avoid making ice. But I mean, fact is that they form the ice, and for some reason, and whether it's really that they want to precipitate out, I don't know. But I mean, fact is, these are the only ice nuclei that make it at such high temperatures, so high, between -10 and 0.
Yeah, but I mean, if you assume that they're doing it for a purpose, then they need to have done that. It must be part of their life cycle, so to say. They need to really be, the cloud phase needs to be part of their life cycle. Otherwise, it's difficult to. I mean, it can be a byprocess of something they normally do. But okay, I'll stop. Qian Wang was the first to raise his hand and ask a question. Qian, you can unmute yourself and ask the question, or.
Oh, should I read it from the chat, or?
I mean, Qian actually raised his hand. And I don't.
Can just ask questions.
He doesn't hear us, maybe. I don't know. So then we have a question from Hans Werner. Now Qian has unmuted. Qian? Okay, I don't hear anything. Test. No. Okay, then I'll take the question from Hans Werner and Hans Werner Jacobi, old colleague of mine with whom I was on Polarstern ages ago.
Former colleague of mine too.
He asked, what about dying microorganisms and their degradation? Is this a significant source of organics for Aerosol-cloud's reign?
It's probably not. I mean, it's some estimates say that biological material can be a significant part of organic aerosols, but that also means dead material. So it can be, so fragments of bacteria cells or anything that used to live can be also, I mean, it's also counted as organic aerosols, and organics are a major part of the aerosol mass. I don't know an exact number, but it's probably definitely that material, of course. It's definitely a part of aerosol and cloud water organics. I mean, it cannot really trace it back. If we find an organic molecule in cloud water, we often just don't know where it comes from, whether it was formed by any chemical reaction or whether it was formed by decay of living material.
Okay, thank you very much. Then we have Subu who raised his hand. Jian, if you get us, send me the chat. Subu,
yeah, sorry. So thanks, Rainer. Barbara, hey, good to see you.
Yeah, hi, Subu.
So yeah, no, I think you actually raised the question I was about to ask, because Christine also mentioned not all biology has a purpose. I'm just wondering how much of the bacteria in the cloud is really on purpose versus accidental, right? If there is fires aloft or there's other material that gets lofted up and bacteria from soil gets, lofted along with it, bacteria didn't mean to be there. And
they make the best out of it, I guess. Yeah.
Yeah, right, exactly. Right.
And that's why they also try to attract water. So to make it as ideal as possible for them.
Right. So I guess it's the second part of my question was basically, do you think there are differences in the bacterial composition across different parts of the world? Like if there's more biofuel cooking, for example, in Nepal, which kind of, in the Himalayas gets lofted up somehow, or if there's more grassland fire burning in Africa, or there's some other soils being lofted up from the wildfires in California, for example, or Australia. So does that lead to different?
Yeah. I mean, I only talked about the large ecosystems, so like deserts in general and seas in general. But of course, if you look on a much smaller scale, it will be very different, even, I don't know on what resolution, but yeah, of course. I mean, it will be very different. And I think the biologists here in the audience know it better than I do, that even soil, the microbial composition in soil can be very different from one location to the next. And it can be on fairly short distances, very different. And I mean, what's in soil will possibly also eventually lift it to the atmosphere.
Right. Great, thanks.
Okay, Thomas raised his hand.
Yes, thanks. Yeah, great talk, very interesting. Makes me think a lot. I was thinking about the typical way we describe aerosols in the atmosphere. We have these satellite data or these profiles from balloons. And what you've shown today is an interesting process of aggregation, which means somehow that when we start with a given profile of aerosols, within the cloud, this aerosol is going to be redistributed in some ways, right? The fine particles are going to aggregate into bigger proteins. So I was wondering how much of this effect can mislead us into starting with a profile in the cloud-free atmosphere that we think is one way and might aggregate and give a very different profile within the cloud. Do you think that that's a major problem, or it's only a minor one?
I don't think so. I mean, the protein molecules are still fairly small. And I think that's something that's happening in one aerosol particle. So it's not that I think that multiple aerosol particles will stick together for some reason. So these are still protein of one bacteria cell, and a single bacteria cell might make up one particle. But what's happening within this particle, it's like chemistry, right? So whether we make one big organic molecule I mean, in the organic aerosol community, there's a lot talk about oligomerization. That's not something you would really distinguish by satellite measurements. I mean, that's the composition of the aerosols, but not the aerosol particle itself that will change.
I see. So it's more the physical property themselves of the bacteria that make it so
Yeah,
special.
it's on a molecular level rather than really on a particle level.
Okay, thanks. Thanks a lot.
Okay, thank you very much.
Hello.
Ah, that's Jen.
Hey.
Okay.
Sorry. So my mic seems having some issue. Anyway, Barbara, it's a very interesting talk. It sounds to me the concentration is still pretty low, so that's why you have to consider the ice nuclear particle as that. But I'm wondering, there must be a strong seasonality, right? So I know the current sample is basically from a water rather than in air. There was no way to measure in air. So, but did you see any very strong seasonality? For example, a order of magnitude increase over a certain season? And
yes,
yeah,
we do have. So obviously, I didn't do the measurements myself, but we do have seasonality, for example, from the pudendome, where we see definitely seasonality in bacteria cell concentration, and probably also bacteria types. But I'm not sure if you have really systematic data there yet, but of course, temperature will affect emissions. And with that will also affect bacteria concentrations. For sure, there's seasonality of different bacteria types from different ecosystems. So there is certainly data, so not only from our group, but also I think also in China, there are quite a few bacteria measurements. And yeah, they show seasonality, of course.
okay. Yeah, another question is, I didn't follow very well on your biodegradation thing, in the influence on the chemical process. I'm just curious, I didn't catch a number. What is the time scale for that degradation? Suppose the cloud is stable, liquid.
Well, the lifetime of a single cloud droplet is about between 10 and 30 minutes somewhere.
Yeah.
But of course, a particle might circulate through a cloud multiple times. So the processing time might be several hours, so in total. So what I showed was only for the processing time of 15 minutes, but we could run our model longer and still get the same ratio between chemistry and biology. So it's not that we really expect that certain compounds will be completely consumed by biodegradation, because also the loss of organic compounds is a lot by deposition, so just removing the particle. But I mean, the overall processing timescale is a few days. So a particle spends several days in the atmosphere. So that would be probably the total time a particle can be processed.
Okay.
Does this answer your question?
Okay. That means if you would take the bacteria perspective, then your story might also change a little, or you look from the like, okay, how much can they grow? What can they get out of their trip in the clouds?
Well, they're always in cloud. So if I say a few days, it's really how long does the particle spend in the atmosphere between emission or formation and until it gets rained out.
Mm-hmm.
But, so
okay.
I mean, it applies to any particle. So really the residence time mostly depends on their size.
Mm-hmm.
I mean, a really large particle will fall down sooner than a very light particle.
Mm-hmm. Okay, yeah, it was more a comment. Sorry. So Subbu, you have raised your hand again.
Yeah, actually, this is made me think, and apologies if we covered this, Barbara, but I mean, is there a I mean, if the cloud droplet, for example, ice, I guess we're talking about ice, but before that, if there's the liquid droplet and there's bacteria in there, and there is some sulfate, some sulfuric acid ions in there, kind of changing the pH of it. Would that affect the properties of the bacteria, or are the concentrations low enough that they're all reasonably externally mixed?
Well, probably it will affect, so probably certain bacteria, I mean, it really depends on the bacteria strain. They can deal better with acidic conditions than other bacteria. So I know that some of the biodegradation measurements that were done in the lab were done at different pH values. I mean, not extreme pH, but something like 3 and 6. So still what we have in clouds. And there was no difference or no significant difference. So they can deal with different pHs. But I guess if it gets really extreme, they might die. So at least some of them might die, some of the cells. Some of them might be able to deal with it better and survive. And probably also depends on the time they are exposed to these extreme pH values. So that's certainly something we also have to consider, that the activity is not a constant. I mean, it will constantly change with the conditions. And that's why I also think if we really want to extend these models and say, well, it's not all happening in the clouds, also in wet aerosols, the activity might be quite different. I mean, the processes might be the same, but the rates might be different. I'm not sure, but I could imagine that some conditions might really affect them.
Thank you.
Okay. I don't see any raised hands anymore. There is another one. Christina, please.
Hi, yeah, I was super fascinated by your talk, Barbara. I had no idea this is what you were doing, so thanks for that. It's really cool stuff, and it strikes me as basically what you're doing is ecosystem ecology in the atmosphere, which is kind of mind-boggling. And I don't really have anything to add other than it seems like it would be really awesome if you could get somebody in one of those positions as a postdoc who came from a more sort of classic ecosystem background, because they could bring a lot of this biology in. I
totally agree. I think a lot of things are known, especially also from the microbiology side.
Exactly, yeah.
Clear to the microbiologists and also to the ecologists, but the atmospheric scientists don't know about it. So
I know. So yeah, I can definitely advertise in my department in Florida, where we have a lot of ecosystem ecologist people and microbial ecology, because I'm really curious about what are the dynamics. Are people studying this? Do they have an idea of basic ecology that's going on in these bacterial communities? Because it seems like you're just describing sort of the tip of the iceberg of some really complex interactions that could have really interesting dynamics, and who knows what the implications are. But it requires like a field of ecology standing behind it, because all of these questions are super complex, right? So doing it on the ground is hard enough.
I mean, I think we are fairly good in describing the transport of aerosol particles, because I mean, that's just physics, right? How big is a particle? How can it be lifted? But what really happens with biological particles, I think there might be a lot known really from the ecology side, but the atmospheric chemists or atmospheric scientists, they start discovering it.
Yeah, really cool.
Yeah, I think I share Christina's excitement. I also think it's absolutely fascinating talk and fascinating field you're working in. Maybe one last thought. Do you think there could be photosynthesis in clouds?
Sorry?
Photosynthesis, so algae living in clouds?
Why not? I mean,
yeah, I mean,
I'm sure there is. I mean, there's enough sunlight. There's water, there's sunlight. I mean, why not?
Yeah, I know from marine science that there are cyanobacteria. They are tiny, like really micrometer range, which could, or nanometer range actually, which are at least small enough to make it into clouds. I don't know if that's happening. Okay, so I have one more: microgardens in clouds. Maybe we get green clouds at some point if we geo-engineer it or something. I don't know if we want that. Okay, I think that's enough for the day. I don't see any more raised hands or questions. It's also 5 o'clock. So yeah, just can again say that was very fascinating, especially for me as a biologist, but probably for everyone to get to know something that they completely had no idea about is taking place. And with that, I would like to close today. Thank you very much for joining. Please